Some of these fundamental landmarks in the evolution of the eukaryotes require us to revisit the tree of life. Traditionally, we've all viewed the tree of life as indeed being a tree with a trunk, branches, and twigs. This here on the right, we have the bacteria at the bottom that gave rise to the protists, that gave rise to the multicellular animals, plants, and fungus. Single common ancestor somewhere down here at the bottom. This more modern version of a phylogeny of all of life, again, implies a single common ancestry somewhere down in here, branches and twigs off to the eubacteria, the archaea, and the eukaryotes. But we now know that the eukaryotes themselves resulted from a merger, that we have mitochondria are now integral to the life of the eukaryotes. That's a separate bacteria that got into an archaebacteria, and then the chloroplasts were cyanobacteria that got into the algae and eventually into the plants. So there are linkages across what we once saw as separate branches. But as we've seen, there are other ways that genes can be transferred from one species to another. When we looked at the evolution of sex, we saw conjugation in bacteria, where one individual may impose its DNA into a recipient. So now we have the selfish DNA of this bacteria present in two individuals. And with transformation, we saw how bacteria will routinely try to reassemble a functioning genome from any freely available bacteria in the environment. We also have seen transduction, and this is where viruses move DNA between hosts. And I want to just go into this in slightly more detail. We saw with genetically modified organisms and genetic engineering how a virus might be used as a vector to deliver a sequence of DNA to a host. This can happen naturally, and you can have bacteriophages that once they get into the host, they take some of the DNA from the bacteria every now and then by accident as they reassemble themselves, and then when they go and they infect a new host, they might be transferring DNA from a previously and now dead host into an entirely new bacteria. That's called transduction. So donor DNA is incorporated into the recipient's chromosome by recombination. So we have many ways that bacteria can have bit of chromosome merged from one species to the next. So we then get what's called lateral gene transfer. Lateral gene transfer is an important concept when we think about antibiotic resistance. This is how resistance can be transferred from one hospital pathogen to another. So if erythromycin is used too heavily in a particular setting, resistance will evolve. And then there could be various mechanisms, whether it's transformation or transduction, that will allow the resistance strain or the resistance genes to erythromycin going from, say, streptococcus to staphylococcus. So we get now the acquisition of a whole gene at once from one unrelated bacteria to another. And resistance in animal pathogens can also do this, so that we could have too much antibiotic use in, say, farms and ranches, and the resistance can develop then in the livestock, and then the bacteria that develop that resistance may cross-infect in humans, and then occasionally the gene for resistance gets transferred into a human form of the bacteria. So antibiotic use in farm animals can lead to resistant strains of a nasty bacteria, E. coli, that then can infect humans. So, given that we can see this in these circumstances of hospitals and in farms, we know it can happen from just very limited metrics of this. Bacteria have been around for billions of years, and so lateral gene transfer must have been common throughout the history of life, even well before the origins of the first eukaryotes. So some people reckon that if you're really serious about trying to describe the history of life in a single tree, it should have lots of cross-linkages between all these different kinds of bacteria, from one eubacteria to another, one archaea to another, maybe from the archaea over the eubacteria, etc., etc. So that the genes that we see in one species of bacteria may actually have been acquired from a very different species. And it turns out that it may be our sense of this single branch giving rise to smaller branches and finer twigs is because multicellular organisms may in fact be the only living things that are unable to merge their genomes with each other. Our large genomes, large numbers of chromosomes are so complex, we cannot freely accept sequences of DNA from other organisms into our germline. And so it is probably a good description of the multicellular organisms that we do have a traditional form. But going back from the origins of life, there must have been a lot of mixture from the very beginning.